Good morning. This is Rajat. I'm an assistant professor in biological engineering from UCT School of Biosystems and Food Engineering. And today I'll be giving a brief overview on risk assessment in a nutshell. So I'll try to finish the entire thing within 40 to 50 minutes. I also set a timer, and then I'll open the floor for discussion. So if you have any question, so you can type in the question and answer section. I'll be happy to address those. So welcome again. And I'm also the chair for Engineers Ireland Agriculture and Food Engineering Division. So towards the end, I'll also request all of you, if you are not member, could you please join our Engineers Ireland group that will actually flourish the dissemination and also the communication between the members and also for the policy makers. So if we want to contribute to any specific problem and what would be the solution, and we can discuss in our division. So welcome. So first thing first, why there's a need for risk assessment. So I actually searched with these keywords in Scopus such as Ireland, environment, human health, pollution, and I found 30 relevant articles. So I created a word cloud with the words in the abstract and in the title. And generally I can found like water, groundwater, aquatic life, air, soil, pesticides, sources, human exposure, antimicrobial resistance, bacteria, metals and metalloids, and ecosystems. So these are the main kind of phrases other than like the main words like Ireland, environment, human health, and pollution. So from the WHO stat, we can find that more than 600 million people worldwide are affected by foodborne pathogens, and like 1 in 10 people fall ill after consumption of contaminated food. And out of the entire disease burden of the world, 25% is contributed from uh, having food which is contaminated with heavy metals and metalloids. So in risk assessment, we have three kinds of fundamental model. The first one is the qualitative model, which is the simplistic one, followed by semi-quantitative model, and then the sophisticated full quantitative model. So in case of qualitative model, we first identify kind of a matrix for impact and probability score. So we try to give a score based on the impact and the probability. The scale can be 1 to 5 or 1 to 10. And then we create kind of equal interval. Say, for example, 1 to 6 is the low score, and then 7 to 12 like medium, and then 13 to 25 is kind of a high risk. And these are very important because, for example, to evaluate any kind of risk for project or any kind of problem, we cannot think multiple things at the same time. So there can be multiple parameters in your problem which can affect the overall outcome. So this actually step helps us to think one parameter at a time. See, for example, your local county council is evaluating two different sites regarding the flood risk, and the key parameters can be likely rainfall, closeness or proximity to the main rivers or streams, type of soil, and the amount of impermeable surfaces. Say these are also very important in terms of comparing multiple sites. So you may have one site which is located in an area subject to occasional rainfall, and then that can be within 1 kilometer from a surface water body. The soil is heavily like clay soil, and there are few impermeable surfaces on the sites. For site 2, it can be located in an area subject to high rainfall. It is 2 kilometers from a main river. The soil is light sandy loam, and there are very few impermeable surfaces on the site. Then calculate the risk score for both sites and compare. So the thing is, you can classify these the sites based on these parameters and with some educated judgment. So you need to have some kind of background knowledge in terms of assessing these sites. For example, first site, the rainfall, the probability score I gave 2, and the impact is 4. The impact is very high. So you can see impact score is kind of same for two sides, because first we need to have the impact. So impact will be the same if we have different kind of probability. And we have like proximity to river, score of 4 for site 1, then type of soil 3. And so because clay can retain water for longer period of time, and the loam or the sandy soil cannot. So the clay soil is more vulnerable to flooding activity. And amount of impermeables means like the water cannot go inside the ground. So the amount of surface water is higher. So in terms of the impermeable surfaces, it will actually help to cause flooding. So we can have a multiplication of probability and impact of each parameter, and then we need to sum it up. So for site 1, we have score of 45. In case of site 2, we have 46. So if I give the same problem to a class of say 50 students, and they may come up with different scores, but towards the end, I can find like the final outcome is kind of comparable. So in our case, site 1 is the winner because we have lower score, but you can see the difference is very minimal. So that's the need of having a kind of further sophisticated model for such kind of problem. So this is the end of the qualitative model. Next, we have semi-quantitative model. So it is kind of sophisticated in terms of the amount of detail we have. So in this case, the fundamental is the same, probability and impact score, but we need to, why this is semi-quantitative? So we can give a score based on certain criteria we set at the beginning, and score can be kind of continuous. So it can be in the form of fraction. So see for the example, so it was my second chapter of my PhD thesis. So anaerobic digestion is a biological process which can utilize farm air, manure, and slurry along with the food waste, and we can produce biogas. However, we need to discard the digestate, which is also a kind of co-product from the process, and it can act as a biofertilizer, and it can provide valuable nutrients to the plants. So we actually sprayed this digestate on agriculture fields. So before I came here, there was a perception in Irish community of farmers that spreading raw farmyard manure and slurry is kind of good or better when we compare with spreading anaerobic digestate. But in reality, after completion of 3 years of my PhD, I found AD is better in terms of biosafety. So I try to score, okay, so the animal manure and slurry contains a lot of bacteria, viruses, and parasites. So we call it pathogen as a whole. And complete inactivation is unlikely through this anaerobic digestion process. So sometimes we need to pasteurize the digestate before the application. So the complete inactivation is unlikely. So we can score this kind of survival from 0 to 1, where like 1 can represent it cannot be inactivated, and 0 means like it can be completely inactivated. So we can use a score, and we can use D value and Z value of individual bacteria for a specific temperature for this pasteurization. So in European context, it is 70 degree for 1 hour, and it was 60 degree for 4 days from the Irish context. But after completion of my PhD, I found 70 degree for 1 hour is sufficient to inactivate these pathogens. So now this is a kind of a part of the legislation. So we just go for that 70 degree and 1 hour. So 60 degree for 4 days is no longer in use. So the first score was in terms of the thermal inactivation, and then score 2 was for the potential transmission pathways. So the bacteria and viruses or parasites can like transmit through air, water, and food, and also animal contact or zoonosis. So I tried to score them for each category as 0.5. So if one pathogen can transmit through all these 4 pathways, it can get a score of 1. And if it can only contaminate through like two pathways, such as air and water, then it would get a score of 0.25 multiplied by 2, that is 0.5. And then the final score, the S3, it was kind of an impact score. So severity or mortality rate in untreated patients. So if you are exposed to this pathogen, you fall ill, and if there is no medication, so what is the chance of getting, a severe illness or death? So the multiplication of these three scores can give the final score for the risk rating, and sometimes probability and impact matrix can be modified. So you can see score 1 and 2, S1 and S2 are the probability score, and S3 is the impact score. So after like getting information from the literature, I found like there are like 91 numbers of pathogens which can present in the contaminated food and infected animals manure and slurry. So out of these 91, because I could not go further with this 91 pathogen for my PhD work, so it is very hard to conduct experiment on all. So I tried to narrow down, so what are the key protagonists or the key pathogen of concern for Ireland? So I found these are Cryptosporidium, Streptococcus, Entamoeba, Salmonella, Ascaris, and so on. So I could rank them. And then I could proceed with the experimental work on each of them along with our project members. So I had 3 PhD friends who are actually part of the entire project. So I designed the experiment from them. So they conducted the lab work, and then I performed the data analysis and came up with the model. So you can see from the semi-quantitative modeling, we can have a kind of clear differentiation between these kind of pathogen of concern, and we can come up with a final decision. So for this semi-quantitative model, if I give the same problem to 50 of my students in the class, they should come up with the same value, not like kind of different value, because it is in the methodology, there is a brief like there is a protocol like this is the process, and you have to follow that procedure to come up with a unique score. So the next one is the most sophisticated risk assessment methodology. We call it like full quantitative or quantitative risk assessment framework. So in case of risk analysis, so it's a 3-step process: risk assessment, risk communication, and risk management. So we manage risk with risk communication. Within the risk assessment, we have 4 sections: hazard identification. So here we actually identify the hazards. So with the semi-quantitative risk ranking approach, we identified the hazards. Then there are like 2 parallel steps: hazard characterization and exposure assessment. So in some paper, I could see the exposure assessment was conducted first and then hazard characterization, and vice versa, because these are independent steps. So in case of hazard characterization, we actually calculate how much of this pathogen, how much of the chemical can cause harm to our body. And in case of exposure assessment, we quantify how much of these pathogens are coming into our body on daily basis. So we quantify the number of bacteria, we calculate the amount of chemicals which coming to our body through different media such as air, water, and food. And combining these 2 steps, hazard characterization and exposure assessment, we actually combine these through the risk characterization step. So it's kind of a bridging step which can combine these 2 and calculate the final risk in a probabilistic way. So 1 in a million, 1 in 100, 1 in 1,000. So that's kind of outcome from the risk characterization. So this is the step for the hazard characterization. So I probably reviewed more than 500 articles to see what are the methodologies for the hazard characterization. So I only found there are only 2 fundamental equations for this. So like the boxing, in boxing we have 3 fundamental moves, if we can recall. So these are, say, jab, hook, and uppercut. So this is kind of simple as that. So we call it exponential model or beta Poisson model. So let me click the pointer. So in case of exponential model, we have the probability can be calculated by this equation 1 minus e to the power minus r multiplied by n. So r is the fitted parameter based on the dose response relationship conducted on like animals, or sometimes we have the outbreak study based on, say for example, in a school, 100 students were given the lunch and 10 found like ill after eating the contaminated food, and then we sample the food, and then if I find the bacteria, with that we back calculate what is the human exposure to those pathogens, and then we can develop the dose response curve. And from that, we calculate the parameter. So that is the statistical parameter R. And N is the number of bacteria we are exposed to on daily basis. So that's the thing we calculate from the exposure assessment part. And then in case of beta Poisson model, we have similar kind of parameter. So it's 1 minus in bracket 1 plus n multiplied by 2 to the power 1 by alpha minus 1, and then by n50, and then 2 to the power of minus alpha. So here the alpha and n50, these are also fitted parameter, and n50 is actually the number of pathogen which can cause 50% illness or infection or like death in the population. So these are called responses, three kinds of responses. And these are different forms of the same equation, equation 3 and 4. So in different paper, I found different kind of format of the same equation, which is equation 2. So the steps are we need to collate the information from the database, because sometimes we cannot conduct the test because of the there are some ethical approval needed, or sometimes it is beyond the scope because the time is limited and these experiments can take a very long time. So we try to develop a kind of a database first, and then try to fit the curve, which can be a collation of all the information into one curve. So the reference original study, or if no, then which is the original study? And then we have the host, like it can be tested on human, it can be tested on rat, mice, dogs, etc. And the best fit model for that data, it can be exponential, it can be beta Poisson model. So out of these equations, we try to type which is the equation used for that specific study. And then these are the parameters. In case of exponential, the R is the statistical parameter. And then route is also important, because sometimes people, the rat can be exposed to this pathogen through eye, and also through gavage, like forcefully feeding. So these routes are also important, because if it is, say for example, a pathogen is going through our body through inhalation, so it would affect the lungs. However, some part of this pathogen, some numbers can slip from the trachea and can go to the stomach. So like ingestion and inhalation pathways should be reported here. It is ideal that if we have the same route of exposure, and the responses is very important to mention, because we cannot compare apple with oranges. So which means like illness can be compared, infection can be compared, or death can be compared with, but like it cannot be compared as illness versus death. So if we have a table of all illnesses and the roots are oral, then we can have multiple studies, and we can actually combine all into one study, one curve, which can be the worst case scenario, and it can capture the dose response relationship from different studies. So then we can use that for our further calculation. So that's part of the chemical hazard characterization. So in case of chemical, so my second project of my PhD was based on the heavy metals. So metals are very important also for development of our cells. So for example, you can see this curve. So so this is kind of the dose, and this is called the response. So you can see death can happen in two scenarios. One is the absence of metals, essential metals, And then this phase is called the deficiency syndrome zone. And then this phase is the healthy state of our health. And then also here we can find the low or mild effects due to the elevated exposure. And then this phase is called the toxic effect. And eventually, if the elevated level is very high, then people can have death as an output or the response. So for a substance, you can see from this table, for a substance, there are three kind of doses from the database: not toxic or beneficial dose, toxic dose, and lethal dose. Lethal dose is corresponding to death. And in case of non-toxic or beneficial dose, you can see there are different kind of examples: alcohol, and then like I think smoking and sleeping pills. So why there are two different terms? So for non-toxic, it is the term used for the compounds such as alcohol and smoking. And the beneficial dose, that's the term associated with medicines. So water can be also toxic if you drink more than say 8 liters per day, because there is a proportion of the salt ions in the blood, which is also important for signaling activity. So if you drink so much of water, that concentration can be reduced and you can have a significant problem. So we can have different doses of the same uh, chemical. So what is called the beneficial dose is a toxic dose, and these are the lethal doses. So what is toxicology? So the study of adverse effects of chemical, physical or biological agents on living organisms and the ecosystem, including the prevention and improvement of such adverse effects. So that's the definition of the toxicity. So toxicity can be influenced by many factors, such as the form of the chemicals, the dosage, and especially the dose time relationship. Like one drug can be administered to you perhaps throughout the day, like 3 times a day, and the same drug can be administered once a day. So in case of once a day, the effects can be harmful. So sometimes the drug is distributed throughout the day. So when the drug is administered, so that is also very important. Exposure route, as I mentioned earlier, it can be ingestion, inhalation, and so on. Species, life stages, such as like infant, young adults, and elderly population. Gender, ability to be absorbed by the body, so that's the most important one. Metabolism, distribution within the body, excretion rate, health of the individual. So if you have underlying illnesses and all, the toxic effect can be higher. So the elderly population and the pregnancy, it's a very crucial aspect in terms of having any kind of pills. So if you go to doctors, so they actually ask these kind of questions first before prescribing. Then the nutritional status of the population, presence of other chemicals, and then circadian rhythms, means like the time of the day the drug is administered. So these are the parameters which can actually affect the toxicity. So the toxicity effects can have one kind of target organ, and sometimes it can affect multiple one. So the name of toxicity can be based on those target organs, such as cardiovascular, then it can be nephrotoxicity, so it is related to nephron, neurotoxicity, that is related to neuron. But sometimes these kind of chemicals may have multiple effects, so we call it systemic toxicity. And these are based on, so these can be categorized further, such as acute toxicity, that is immediately. So if I'm exposed to any harmful chemicals or poisons, so I may have immediate death. So that is called the acute toxicity. Then we have subchronic toxicity. It can take several weeks or months to develop. Chronic toxicity, it can take many months or years. And then we have the carcinogenicity, so that leads to the cancer. Developmental toxicity, that is in the form of embryo, so from mother to unborn child. Then we have genetic toxicity, that is related to the DNA damage. So it can be the change of the sequence of the DNA, changes in the chromosomal structure, and increase or decrease the number of chromosomes. Next, we can see the same toxic chemical may have different kind of toxicity when we consider the acute or the chronic. Say, for example, the ethanol, for the acute toxicity, it can damage the central nervous system. But in terms of the chronic toxicity with the lower dose, it can cause liver cancer. So that is same with arsenic. For the acute, with the higher dose, it can cause gastrointestinal damage, and for the chronic exposure, it can cause skin or liver cancer. So these are the steps of the toxicity. We start with toxicity for the cell lines. Cell lines can be different cells from our body. So it can be epithelial cells, and there are many cell lines. So you can have a look later on. So we can actually do the test in the lab. And then we have the toxicity on Daphnia magna. So that is a kind of waterfly. So we can test them for different chemicals for the toxicity, and it's called the Daphnia immobile test. So if it is toxic, they do not move much. So based on that, we determine the ecotoxicity, or sometimes it's called EC50. So the dose responsible to cause toxic effect 50% on Daphnia. After that, so one chemical, it passes the cytotoxicity, then we need to conduct the toxicity on Daphnia, and then if passes that, then we move forward to the fish and then rodent models such as rat and rabbits. Then we can perform the test on dogs and human. So most of the time, the pills or the drugs are tested on rats or the rodents, because we are mammals, so we can actually extrapolate from these results. And also for the chronic test, I think we need to go for a test on dog. So that was the challenge, because for chronic test we need to wait for perhaps like years, 1 or 2 years, or maybe several years. So that was the challenge during the COVID time for development of the drug, because we couldn't stay for that much long for the toxicity test. And it was so these are some also situation when we need to take some decision, because if we do not take this medicine, maybe you can be you can have severe problem due to this virus. But if you consider these drugs, there can be some long-term toxic effect on you. So for the betterment of the population, sometimes we actually approve certain drugs for the betterment of the humanity. So now this is it is kind of intense, these two slides, but it is my responsibility to disclose the new updates on the hazard characterization methods, because from 2023, I think, IFSA proposed a new methodology, and the health-based guideline values sometimes it's not used anymore. So this is kind of a history, how the toxicity or the safe limits of different chemicals were developed, and then I will explain you the most recent update on this test. So after conducting tests on different animals and all, we need to identify these two levels. So NOEL and LOEL. So no means no observed adverse effect level on mice or rats. And this is kind of lowest observed adverse level on different animals. So this is dose, x-axis, and this is the response in the y-axis. Means no effect, means like we are increasing the dose from low to high, and we need to identify at which, up to which dose there is no such kind of adverse effect. So once we know the NOEL, then we need to divide that by different uncertainty factors to calculate the reference dose, or RFD. So these are the uncertainty factors. So uncertainty factors can arise from the human variability. So if you are conducting that test on any human, so human from say European population, so the people living in Asia may have different response. So we have the variability. Then we have the variability based on different animals, And then so it can be within the study and across the study. And then 10 for the less than chronic exposure. So sometimes if you conduct test on only acute kind of effect, you are not considering that for 2 years and so on. So then you need to consider that uncertainty factor as 10. And then the last one is if we know the Lowell instead of Noel, and then we need to use this uncertainty factor of 10. But I have seen in many papers like people use Noel value, but still they divide the entire thing by these 4 uncertain factors. But that is wrong. If we have Noel, you need to divide the entire thing by 3 uncertainty factors, 10, 10, and 10. But if you have the Lowell value, then only you need to divide that by 4 uncertain factors. So that is 10 to the power 4. So once we have this RFD derived from this Noel value, so then we can actually convert that to different kinds of recommendations, such as weekly allowable intake or daily allowable intake, something like that. So then this is a important term such as hazard quotient. So hazard quotient is the estimation of health risk exposure to chemical pollutants. It can indicate like if the value is more than 1, so there is a potential risk but not confirmatory, because then we need to come up with the long-term effect such as target hazard quotient. So that can look at the different kind of hazards throughout the year, or maybe multiple year, or throughout the lifetime of people. And basically how we can calculate this hazard quotient. So for example, if I consume 70 grams of potatoes, and then if I know the concentration of the heavy metals or metalloids in the potato, so that is the multiplication divided by my body mass. So if my weight is 70 kg, so I need to divide that by my weight, and then I need to divide the RFD, so that is derived from this health-based guideline value, so which is again based on the Noel or Lowell value. So if I know that values, then I can calculate the hazard quotient for myself, and if it is less than 1, then it's fine. So that is kind of, I'm safe, but still I need to conduct that THQ test for long term. But if it is more than 1, then there is a potential risk and further investigation is needed in terms of, so there is kind of an alert that it is more than 1. So definitely there will be a problem in terms of long term, right? So if it is less than 1, still there's a chance that it can be toxic in the long term. But if it is more than 1, so definitely you will be sure that I have a problem in the future. I will have some problem in the future. So now, so that's the most sophisticated model, which was introduced in 2023 by EFSA. So this is kind of an alteration from the previous one, but not a completely kind of new methodology. So here, what we do, we also do this kind of dose response kind of plot, where this is from low to high dose in x-axis, and we have the responses. So we plot, so we actually conduct this kind of test on different animals, and we have the confidence interval, means like the 55 percentile and 95 percentile, the values of the responses with the mean value. So if I connect the dots of all these means, so I generally have a S-shaped curve. So that is the basic dose response curve. So this part is linear, this part is exponential, and this part is also linear. So we call that part as low dose linearity. Anyway, so we can extrapolate that line to the y-axis, and then that's my benchmark response. And this benchmark response, the difference between this and increased by 5%. So we can increase this response by 5% or 10%. So based on that, we actually call this BMDL as 5% or 10%. Anyway, so we actually increase that by 5% for the factor of safety, and we find this point, and then we draw a straight line from here, which is horizontal to x-axis, And the intersection points of this 95th percentile and the 5th percentile we record. So the corresponding value of the x-axis, so that is called the BMDL. So benchmark dose lower, and for this case we need to call it BMDL 5. So if it was 10%, we should call it BMDL 10. So now this value, when it touches this 5th percentile, this straight line, and the corresponding x value, so that's your BMD upper. So now we have two scenarios. If the chemical is carcinogenic or genotoxic, or if the chemical is non-carcinogenic and non-genotoxic. If it is non-genotoxic and non-carcinogenic, so the margin of exposure should be calculated as Noel value divided by estimated exposure dose. So estimated exposure dose is we calculate through the exposure assessment. So the amount of chemical I'm exposed to on daily basis through that pathway. So it can be oral, it can be inhalation, and so on. So if this margin of exposure is equal or higher than 100, then we are safe. Okay. But in case of some chemicals which are genotoxic and carcinogenic, we do not have actually the Noel value, because the curve will start from here. So it will start from here, because at very small dose also it can cause problem in long term. So it can be genotoxic and carcinogenic in the long term. So that's why we identified that BMDL lower, like this value, and divide by the estimated exposure dose. So that is our margin of exposure. So there's slight difference from this Noel value and the BMDL. So BMDL is the one we use for this margin of exposure for genotoxic and carcinogenic compounds. So it should be equal or higher than 10 to the power 4. So why 10 to the power 4? Because that's a total uncertainty I presented in the past. So there can be 10 uncertainty factor for 4 factors. So it should be more than 10 to the power 4. And then we can call that compound as safe. So these are some comparison. So I'll actually share the slides after this talk, and you can have a read for the comparison of advantages and disadvantages of this BMDL and then the Noel methodology. Next, we have the exposure assessment. This is also atypical. So I found it is important to give you some example instead of just the theory. So here in this stage of risk assessment, we quantify the number of pathogen or the amount of chemical we are exposed to on daily basis. So for example, my student was doing the risk assessment of using reclaimed irrigation water for irrigation purpose, for the sprinkler irrigation to grow different crops. And after the harvest of these crops, it can be washed with or cleaned with chlorinated water, and it can be packaged and it can be kept at cold temperature and we can eat. So the pathogen propagation can be from this to this. Okay. So initial pathogen concentration in untreated irrigation water was monitored and calculated from the lab experiment, and then we predicted what would be the attachment if we use the sprinkler irrigation for such crops. And then this attachment can be here on top of these leaves. However, the sunlight can inactivate a lot of pathogen due to the presence of ultraviolet ray and the heat. So after the addition of these pathogen on top of these leaves, so it undergoes decay. And then after the harvest, we actually inactivate a lot of pathogen with this chlorine. And then after we place the ready-to-eat salad on shelves, some bacteria can grow also at that temperature. So generally we have the shelf life of say 1 week, and the temperature is kind of 4 to 7 degrees on the shelf. So some bacteria, not all, can grow at this stage, but not in a huge quantity. And then when we consume our salads, so average European consumes 35 grams of salad. And with that, we actually calculate the number of pathogen we can be exposed to. It looks like scary, but I'll present the results soon, and you will find it is not that much scary, and that's why we eat salad and we do not generally, fall ill after consumption of these salads. So the next stage is called risk characterization. So in case of risk characterization, we combine the hazard characterization and exposure assessment steps. So for the deterministic model, so we actually consider the mean value of this food intake, and then presence or the probability, we consider it is present all the time, and then we calculate the concentration of different chemicals or pathogen in the food and we multiply. However, this deterministic approach is not sophisticated because there can be a lot of uncertainties. So the model uncertainty can be of these kinds. Parts of pathway can be unknown and several alternatives can be there. In terms of input uncertainty, true value of the input is unknown and limited amount of information, and it reflects the degree of knowledge of the people who are conducting the test. Say, for example, if I need to sample the water from a lake to calculate the amount of bacteria in the water, so I can sample from many parts of that lake. So I can come up with different values of the bacteria levels. So these are called variability because this is naturally occurring. But if I send that sample to 5 different labs and based on the skill of the people who are actually conducting those instruments and also based on the sophisticated nature of those instruments, we can have different values. So these are called uncertainty. So the total uncertainty is the sum of variability and uncertainty. So these are the difference between the variability and uncertainty. And what we do in the probabilistic approach, we have some input parameters and we multiply or divide, or in the model we use different functions and we actually predict the output. So output is a distribution, not a fixed value. So these are some results of my students. So you can see the first 2 columns. These are actually human exposure, these first 2 columns. So these are the number of pathogen people can be exposed to on daily basis. So you can see this is the simulated mean value and these are the confidence interval like 5th and 95th percentile. So you can see total coliform. So these are fine because for those total coliform, indicator E. coli and enterococcus or enterococci, we do not have dose response model because these are not pathogenic. However, these are indicator bacteria. We quantify these to surrogate the for the other bacteria of concern. So if they are not present, we can actually predict that the other pathogen may not be present. So that's why we call them as indicator. So we can stop calculation at this end for these 3 bacteria of concern: total coliform, E. coli, indicator bacteria, and enterococci. So you can see these values are less than 1. So that is fine because the legislation says the value should be less than 1 in 100 ml of water. However, you can see for the 95th percentile, okay, so that is also less than 1. So that is fine. But in some cases, in my like experiment for my project, I found that is less than 2. So 95th percentile is important because that is the upper threshold of this uncertainty. So we need to actually calculate and sometimes take decision based on that 95th percentile. So the 5th percentile is less than the simulated mean, and this 95th percentile is always higher than the simulated mean. And then for other pathogen of concern, we found the annual risk. So we calculate the daily risk from the dose response model, and with this we use this equation: P annual equals to 1 minus P daily to the power of number of days of consumption. So in the worst case scenario, if I say a person is eating salad every day throughout the year, so that is 365, And it can be different also. If you say I only eat 20 days in a year, so it can be 20 for that. So we can actually extrapolate from daily risk to annual risk. And then we need to compare with WHO limit. So 1 in a million is a kind of a gold standard for chemical risk, and the endpoint is death as per WHO. But for microbial risk, the endpoint is sometimes illness or infection. And for that illness, we have a threshold of 10 to the power 1 in 10 to the power 4 instead of 1 in 10 to the power 6 or the million. So then we can compare the annual risk based on this kind of threshold and take a decision whether it is low risk, moderate risk, or high risk. And then you can see that properly here. So we try to rank these pathogen of concern. So you can see this is the threshold limit, and our values are below that threshold limit. So there is low risk associated with this pathogen of concern. However, you can rank them, such as based on the risk of pathogen based on the illness. You can see Shigella, Cryptosporidium, Salmonella, Giardia, Clostridium, and E. coli pathogenic. So pathogenic and non-pathogenic in total. However, for the infection, so as I mentioned you earlier, in the hazard characterization, the endpoint is important, whether it is infection, illness, or death. So we found some equation for the infection, and these are Campylobacter, norovirus, adenovirus, and rotavirus. So the viruses are, most of them are here, which can cause infection. So first we develop the illness, then infection, and then death. So that's the stages of the responses. Then we perform the sensitivity analysis to see the which of these parameter, because this is the probabilistic model, so it's not a deterministic one. So which parameter may have the influence of our model. So positive correlation means, so things are positively correlated. Negative means negative correlation. See, for example, we found the consumption of the salads is kind of the most influential parameter which can be positively correlated. And then we have the decay constant, which is negatively correlated. And because, which means if we have high decay, we can ensure that our risk can be minimal, because with the decay, a lot of pathogen can be dead. And then we have the initial concentration, which makes sense. If we have the initial concentration high in the irrigated water, then we have the higher risk, right? So next we have the combination of chemical and microbial risk. So I gave the example of my student's work because his main objective was to calculate the benefit risk assessment of using chlorine for this treatment. Because as I mentioned earlier, the residual chlorine or any chemical can be toxic at certain point, certain level or the concentration. So we ran two scenarios. So let me talk about the DALI first. What is disability adjusted life years? So which means if the life expectancy of Irish population is say 80 years, and if I get cancer at the age of 65, then perhaps I can live for another 2 years, but I would live that 2 years with disability, right? So between 65 and 67, and then eventually I die. So the number of years lost from the average expectancy of Irish population, so that is 80 years. So that is the term called year lost. So this is a kind of simple example. So if you are exposed to this kind of pathogen, you may have some infection or illness. So based on that, the year lost from your life we can calculate. And if you are exposed to some chemicals, we can calculate the margin of exposure. And with the margin of exposure, the life expectancy, or we can actually predict how much amount of year can be lost from my life. And then this can act, that DALI can act as a common currency for this comparison, because we cannot compare the microbial risk to the chemical risk otherwise. So in the baseline scenario, we use this chlorinated water to disinfect the salads before packaging, and then we quantified the annual risk from this pathogen and the annual risk from this chemical, so microbial and chemical risk. In the second scenario, where no chlorine is applied, and then we calculated the quantitative microbial risk assessment only, no chemical risk, so that's only the microbial for the DALI, and then we compare these two DALI. So here, this is the results. So for the first scenario, so DALI is 0.9 years in 100,000 population. So in the second case, we have 1.1 years in 100,000 population. So you can see here, for the baseline scenario, the number of years lost for the population is less than this one. So that is a good practice to use chlorinated water with very low dose. So this chlorinated means like you cannot actually detect that through the taste and all. So only through the lab test we can actually detect the residual chlorine level, and that should be below the threshold limit set by EPA or Food Safety Authority. So that's the need of comparing this different chemical and microbial risk with DALI. So I'm now towards the end of this presentation. So this is kind of my scenario of my current research. So this is the extended collaborative network. So you can see my research profile from our university website. So from the dimension of well-being, so there are many aspects. I only focus on the physical and environmental aspects through my research. So I look at the air, water, food, and the zoonotic, like the contact for different pathogens, and also for heavy metals and metalloids problem in Ireland. So Q Omega is my predominant strength kind of in terms of my research. Then I also conduct the chemical risk for pesticides, then nanoparticles, good nanoparticles or engineered nanoparticles used for food, then micro and nanoplastics, and then benefit risk assessment. Say, for example, the coating on your frying pan. So if you do not have coating on your frying pan, then you are consuming more oil, which can be bad in terms of bad cholesterol and all. But if you use those kind of coatings, sometimes for the prolonged exposure, the leaching from these coating can actually have some problem in your body in terms of chemical risk, because nano coating is applied on top of these frying pans. And if you see the Danish chemical risk assessment database, you will see like there are many such coatings are banned in terms of this frying pan. And also I see like the projection of different risk in terms of chemical or microbial risk under the climate change scenarios. So these are some completed projects of mine. So quantitative microbial risk assessment of spreading anaerobic digestate on land, human health risk assessment of heavy metals and metalloids. These two are part of my PhD. Then as a postdoc, I performed the environmental and human health risk assessment of pathogen trapping protein solutions. So those proteins actually trapped the fish pathogen in the water, and they can be sedimented at the bottom so that the fish will not be given any antibiotics. And what would be the residual kind of risk from those proteins? So that was the objective of that project. And then I was mentoring one a fellow for the antibiotic resistant bacteria. And then from the co-supervision point of view, bisphenol A, mycotoxins, micro and nanoplastics, data-driven model, machine learning, and then the climate change scenarios. Then like ready-to-eat salads. These are the projects I handled in the past, and I'm actually supervising two students at the moment on PFAS and groundwater contamination, and also some with the nanoplastics. So there is one conference also. I'm taking advantage of this presentation to invite you for a national conference. So Environment is the largest national conference on environment. So UCD is hosting this conference after 12 years. So I'm the host. And please go through this website, and if you're interested, join or register yourself for this conference. And these are the themes you may have interest in. And right now we have more than 300 registrations for this conference. And if you are interested, also please keep in touch. And being a member of Engineers Ireland, if you're interested in agriculture and food division, so please get in touch with us. And we need a lot of more new members in our division to flourish our aims and objectives. So I'd like to thank finally Professor Enda Cummins. He was my supervisor for my PhD, and he's a lifelong mentor for my research and teaching work. And the Director General of Engineers Ireland, Pat, and also Sinead, and all the members of Agriculture and Food Division. Thank you very much.